Good day. I am Brian Hoffland, President of Research to Prevent Blindness. Welcome to RPB's Lunch and Learn, Eye on Geographic Atrophy and Dry AMD. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. RPB was founded in 1960, and over our 62-year history, we have awarded more than $393 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. In today's educational session, you will hear from two RPB-supported talented scientists about geographic atrophy and dry AMD and their causes, impact, and cutting-edge research. One of our speakers, Dr. Srinivas Sada, received an RPB Physician Scientist Award, and the other speaker, Dr. Eleanor Ladd, received an RPB Special Scholar Award. Thank you to all of the generous supporters of RPB who make our work possible. I'd especially like to thank Apellis Pharmaceuticals for their support of today's educational event. The importance of supporting medical research has never been so apparent. We thank you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. If you have not yet supported RPB, please consider doing so to help make sure we are able to continue these breakthroughs. But getting back to today's session, let me introduce our moderator. Ramiro Ribeiro, MD, PhD, is the Executive Medical Director and Ophthalmology Program Lead at Apellis Pharmaceuticals, which develops transformative therapies for people living with a broad range of debilitating diseases. Apellis treatments are designed to control complement, part of the body's immune system. Dr. Ribeiro has worked for Apellis since 2018. A scientist and an ophthalmologist by training, Dr. Ribeiro continues his areas of expertise to lead Apellis's clinical development of the phase 3 program in geographic atrophy, secondary to AMD. Previously, he held many leadership roles in drug development for private companies. And before turning his talents to drug developments, Dr. Ribeiro was a practicing ophthalmologist in Brazil, performing cataract, retinal, and glaucoma surgeries. Dr. Ribeiro will introduce the two speakers for today, and he will moderate the Q&A session at the end. Each speaker will speak for 20 minutes, and I will be the timekeeper. I will give them a warning at the 18-minute mark. We want to make sure that we have ample time at the end for a robust Q&A. Dr. Ribeiro, please take it away.
Thanks so much, Brian. And I'm very excited to be here. As Brian mentioned, our event today is on geographic atrophy and dry age-related macular degeneration. I'm very excited for today's discussion during the AMD Awareness Month, especially now that we're very close than ever to potentially get a therapy that is FDA approved. As Brent mentioned, I'm the clinical lead for the GA program at Apellis. Here at Apellis, we are committed to delivering therapy for patients with complement-driven disease, which we believe include AMD and GA. We have recently announced the results from our phase 3 Derby and Oak studies in geographic atrophy, and we are working really hard to potentially bring this therapy to patients. With that, I want to present our first speaker, Dr. Sada, which will give us an overview of AMD and GA. Dr. Sada is Director of Artificial Intelligence and Imaging Research at the Doheny Eye Institute and Professor of Ophthalmology at the University of California, Los Angeles, UCLA, Gaffney School of Medicine. Dr. Sada received his MD from Johns Hopkins University, where he also completed ophthalmology residency and neuro-ophthalmology and medical retina fellowships. He served as an assistant professor of ophthalmology at Johns Hopkins before being recruited to Doheny. Dr. Sada's major research interests include retina image analysis, advanced retina imaging technologies, and clinical trial endpoint design. He has more than 600 peer-reviewed publications and 20 book chapters, and has given over 450 presentations worldwide. Vas, it's a pleasure to have you here.
Thanks so much, Ramiro. And I also wanted to thank Research to Prevent Blindness for the opportunity to present at the symposium, but also for their support over the years, not only for my research, but really supporting research in the field. And as Brian highlighted earlier, this is really what allows breakthroughs to occur. So for that, I'm very grateful. So I'm really pleased to, again, be able to participate in this symposium and to talk about AMD, and in particular, geographic atrophy, which I think we'll all agree is a very important topic for discussion. Now, I know that in our audience, there are people who are very familiar with the eye, but also people who may not be so familiar with the detailed anatomy of the eye and specifically the macula. So I thought I would start with some basics, really to try to get us all on the same page as we sort of go through some of the more detailed concepts of where the state of the art in the field currently lies. So when I talk about the eye just in general and give these types of presentations, and even when patients come to us in the clinic, people are sometimes surprised to hear that we have so many different subspecialties in ophthalmology, cornea specialists, retina specialists, and you think, how can that be? It's such a small organ. And while it's a small organ, it's an incredibly complex one as well, but it can be kind of broken down into two parts. You can envision the eye as being composed of a sort of a front part, which is kind of the focusing system. It contains a fixed focus lens in the cornea, as well as an adjustable focus lens that allows you to focus at distance to near, and it focuses the information onto the back part of the eye where the retina lies. And that contains the special light sensitive cells that then send the information to the brain via the optic nerve. So that's how vision occurs. Now, this is one of the reasons why oftentimes when I talk about the eye, and especially discuss it with patients, I like to use the eye camera analogy, because in many ways they operate on similar principles. And of course, in the eye, the retina is the equivalent to the film of the camera, again, containing those special cells that allow light to be converted into visual information. And so when we're talking about the macula, then we're talking about a very small part of the retina, a very important part of the retina. So if you can imagine, again, to extend this film to retina analogy, and think about the retina as the camera film, think about your peripheral retina as kind of like a 1 megapixel camera, but the macula is kind of like a 40 megapixel camera. It's what we need for the very fine vision that we have with respect to our central vision. And of course, this central vision is very important for many of the tasks that we do in our daily life that I'll talk about. Now, even though I use this retina as the film of the camera analogy, and it is a very thin structure, it's in general less than 1/3 of a millimeter thick, it itself is composed of many different layers of cells. And so this is sort of what we see under the microscope, but then there's a cartoon schematic sort of showing that there's a complex layering and interactions between a number of different cells. And in fact, at the very bottom of the retina, we have these specialized cells we call rods and cones. They're the light sensitive cells or photoreceptors, and then they transmit their information to a series of other processing cells that allow vision to occur. I also want to highlight, and this is going to be very important for the rest of the presentation and what you're going to hear from myself and Dr. Ladd, is that there are also this layer of cells at the very bottom, sort of underneath the retina, if you will, which we call the retinal pigment epithelium. These are very special cells that support the retina. They actually turn out to be the most metabolically active cells in the body. And so not surprisingly, they have a blood supply directly below them that really tries to provide the energy required to support these cells as well as the overlying retina. So that's kind of a quick snapshot to orient you to this very complex structure in the eye and in the retina and in the macula in particular that allows all of us to be able to see. So age-related macular degeneration, or AMD, as the name applies, or would seem to describe, is a condition that impacts the macula. Okay, so or the center part of the retina, and obviously we're talking about a degeneration that's occurring to this structure. So I thought it's also important to highlight some basic facts about AMD and why I think all of us care. And one of the reasons we should care is that it's the leading cause of irreversible blindness in older individuals, and really the leading cause of blindness still in the United States. And one of the reasons is because it's common. We often say that 30% of individuals over the age of 65 will develop this. And I would actually argue that if we all live to an old enough age, if we all could live to 120, I think nearly all of us would have evidence of macular degeneration. And it's also something that's more common in women. Part of this may be related to the fact that women tend to live longer than men. As I mentioned, because this condition affects the macula, it should come as no surprise that the part of our vision that's most severely affected is our central vision. So the peripheral vision is relatively spared in this condition. So what's at threat is losing our central vision, which obviously is super important for reading, driving, watching television, all of these types of activities. So if macular degeneration gets severe, it's something that can lead to legal blindness, although in general, it's not something that would make one go completely blind because your peripheral vision is relatively spared. At the same time, though, it is worth pointing out that that night vision can also be impacted in patients with macular degeneration. It's oftentimes a frequent complaint of people who have macular degeneration. So I want to highlight that even though this is primarily a macular disease, there certainly can be an effect on the rods and some aspects of the peripheral retina as well. So how does the doctor make the diagnosis of AMD? How do we figure out that someone has AMD? Well, we look for abnormalities affecting the macula. In particular, we look for the accumulation early on of a material or an abnormality we call drusen. And this is really the accumulation of fat and proteins underneath the retina. So this is sort of a close-up of a portion of that cartoon I showed earlier. And these are the rods and cones. This is that layer of RPE cells, this is the layer of blood vessels below. And drusen essentially are material that accumulate in this area, essentially below that layer of RPE or retinal pigment epithelial cells. And again, you don't have to be a retina expert to see the abnormality. And again, this is the appearance of the normal macula when we look in and examine a patient's eye. And again, it's very easy to see this accumulation of these fats and proteins and immune deposits that accumulate and underneath the retina in a patient who develops signs of macular degeneration. You oftentimes hear these terms dry versus wet macular degeneration. What do we mean by that? So when we just have the deposits, we might call it as being dry. Well, what's wet macular degeneration? Well, by virtue of these deposits being there, these drusen deposits, that can cause eventually some injury to the structures that separate the retina from these layers of blood vessels. And this can incite and allow the blood vessels to grow through these cracks and ultimately grow directly underneath the RPE and the retina. And of course, these blood vessels, new blood vessels tend to be fragile. We call these new blood vessels neo or new vascularization. And these new blood vessels can bleed and leak. And you can imagine that if you get this kind of bleeding, that's probably not something that's good for the retinal photoreceptors. So we call that wet macular degeneration. The blood, the serum, all these leaking components, they can cause damage to those sensitive cells and then ultimately can lead, if untreated, to these kinds of large scars as the blood and the other damage starts to heal. Unfortunately, that scarring results in loss of those light-sensitive cells and loss of vision. Now, obviously, when you're eye doctor, when we look inside the eye, as retina specialists, we can see these areas of bleeding, and it's relatively easy to make the diagnosis. We also have specialized imaging tools. So one of the advantages we have as ophthalmologists is that because light has to get into the eye, that means we can see into the eye to do this kind of examination. But we can also use take pictures of the eye in that same type of way. And one of the tools that we use is something we call optical coherence tomography, or OCT. It allows us to see actually those different layers of the retina. So you might imagine, many of you are familiar with CT, MRI. Those are also tools to be able to see this kind of detail, these kinds of almost sections through tissue, almost like what you'd see under a microscope. But the beauty in the eye is that we can use light-based techniques because the eye is relatively transparent. And again, these are the normal layers of the retina in a normal eye. And I want to highlight again, you don't have to be a retina specialist to recognize this is quite different. So this is all that bleeding and the like that's elevated the retina and caused disruption to the function of the retinal cells in this patient with wet macular degeneration. Now, there are other tools that as eye doctors we often use to diagnose the presence of wet macular degeneration. We can actually inject a dye through the vein. This is actually, this fluorescein dye is the same dye that used to be used to color the Chicago River green on St. Patrick's Day. But in any event, it allows us to highlight areas of abnormality. So you can see, again, compared to a normal eye, you can see this unusual area of leakage of the dye related to the development of new blood vessels. And that's one of the tools that we use to diagnose wet macular degeneration. Nowadays, we even have advances in our OCT technology where we're able to start to see the vessels associated with these areas of new blood vessel growth from wet macular degeneration. And we're able to do that even without injecting dye, which is kind of an amazing achievement that's been possible in the last few years. So I talked a little bit about why we think these new vessels may develop, and there's been a lot of new science around this. It's an area certainly that's been of a lot of personal interest to myself. One concept is that these drusen deposits may be a sign of a struggling retina, and these new blood vessels that appear may be the last-ditch effort by the body to try to bring more nutrition and save these cells. And so sometimes even when I talk to patients about wet macular degeneration, I say, well, it's kind of a good idea, but it's poorly executed because these new blood vessels then can leak and bleed and cause more damage. The good news is that we've had a lot of progress over the last couple of decades in treating wet macular degeneration. We can inject medications that are able to stop blood vessel growth and maybe even reverse the growth of these blood vessels, which has been a great achievement compared to when I trained to what we can do now. We can actually stabilize vision and improve it in many cases in these patients. The bad news, though, is these injections don't last very long, so they have to be repeated frequently, sometimes every month. But maybe even the worst news, and I call this the good, bad, and the ugly, is that it doesn't do anything about the dry macular degeneration. You can still lose vision, as I'll highlight, due to loss of those light-sensitive cells, even if you're able to control the wet macular degeneration. Now, we've had some progress in how we do it. There are longer acting drugs, maybe even devices that we can use, almost like little implants in your eye they can refill so they can release the drugs over time. And that can be very helpful so you don't have to have an injection every month, and there may be gene therapy opportunities as well. But the bottom line is that it doesn't do anything about the dry macular degeneration and the progression to atrophy. And so even with or without the development of these blood vessels, patients can progress to develop atrophy. Again, you can see the change in this patient has evidence of those drusen deposits, but you can see that that's changed over time. And what you're seeing here is actually a loss of the light-sensitive cells. Again, maybe it's easier to see when you compare normal macula to a macula with geographic atrophy. And that really is, when we talk about geographic atrophy, we're talking about loss of the RPE cells as well as the rods and cones, the light-sensitive cells overlying them. And you can imagine that if you have a big hole like that where you don't have those light-sensitive cells, that's going to be a problem with respect to vision. You can also see it on those cross-sectional OCT images. You can see in this case a drusen deposit, and you can see over time it collapses and you can start to see loss of the layers of light-sensitive cells. This band here happens to correspond to those rods and cones, and you can see how it's missing here. And you can imagine how that could be associated with loss of vision as you progress to atrophy. And again, this is what a patient might notice, where you might see essentially missing areas in your vision or a blurred spot, and that can be really problematic for many of the activities that are important in our lives. So, so one of the things I want to highlight, though, is that, of course, you can see atrophy or geographic atrophy in a patient who just progresses from dry macular degeneration, continues to progress and lose these light-sensitive cells. But even with a patient who develops wet macular degeneration, when we treat it, yes, we've got these great treatments that can make the bleeding and the fluid stop. But patients can still, unfortunately, lose vision because we haven't done anything about controlling this atrophy process. And that's one of the reasons why in the past we used to think about, people would come in, they ask me, do I have wet AMD or dry AMD? And I have to say that we don't really think about the disease in this very binary way anymore. We think about AMD as a disease. We first see it with the start of drusen and ultimately can progress to atrophy. And we think now that wet AMD might be something that occurs in some patients, maybe that last ditch effort to try to save the retinal cells, and we can treat that. but unfortunately, these patients can still develop atrophy. So atrophy is what we really need to stop. Dr. Ladd in her presentation is going to talk about some of the exciting advances and how we may have opportunities to do something about atrophy. But so far, it seems that the best we can do is stop this kind of progressive enlargement of the loss of retinal cells. It's easy to see on this black and white image, this area. This is a special kind of technology called fundus autofluorescence, where you can see this loss of light-sensitive cells. But I wanted to highlight that the best we can do is maybe slow the enlargement. We can't currently stop it or reverse it. And that's obviously something that we want to tackle going forward. You might ask, well, why has it been so difficult? Why is it so hard to stop this atrophy process? And there's been some concern, maybe, could we be treating too late in the disease process? Is the horse already left the barn? And so we really want to focus, I think, going forward in the future on how can we intervene even earlier. And if we're going to treat earlier, we need to identify features we call biomarkers that may be associated with a higher risk of progression to advanced stage. So we really want to know, if we don't treat a patient, who's going to progress to atrophy in 2 years as opposed to 20 years? Because obviously somebody who's going to progress fast is maybe the kind of person we really want to focus on in terms of treating early. And so we've been, a focus of my own research personally has been in hunting for these kinds of biomarkers. Specifically, I've been interested in hunting for them on our OCT type scans. And again, you can see two different patients with AMD. And I think you can recognize, even not being a retina specialist, both of them have these bumps related to the drusen, but this patient also has these bright spots in the retina. And I'll talk a little bit about that next as I finish up on this presentation. And so the good news is, over the years, we've identified a number of different biomarkers or factors that seem to be associated with a higher risk for progression. And you can imagine that, it probably makes a difference if you just have a few of these bumps, few of these drusen, versus if you have a lot of them. And we've been able to show that patients who have a lot more of these drusen, they seem to be at higher risk for progressing to atrophy. Also, we can see that not all drusen are the same. Sometimes drusen can develop little dark spots within them. That can be a sign of maybe, that seems to be a sign of some calcium buildup, which could be a sign of an RPE cell or a retina that's struggling. Another sign of a struggling retina can be if the RPE cells actually leave their normal location and go into the retina itself. Again, a sign of an eye that might be in trouble. So that can be a biomarker that can help us in identifying patients who be at risk. We've also identified that drusen-like material can not only accumulate below the RPE, but can accumulate above the RPE as well, something we call subretinal drusenoid deposits or reticular pseudodrusen. All these names aren't important as much as to highlight that we're identifying these types of features that highlight patients who may be in trouble. And in fact, we've also—
You should wrap up in the next 2 minutes, please.
Yep, I will. And of course, now we're able to identify even biomarkers of patients who may be just on the verge of developing atrophy, something we call incomplete atrophy. And we've shown in a variety of studies that these factors seem to be associated with a higher risk for progression to advanced AMD. The exciting thing now is, of course, we can recognize them looking at these images, but can we do them in a more automatic fashion? And that's where, in this era of artificial intelligence, where we can teach essentially machines to actually identify these biomarkers automatically, we've been able to make a dramatic progress. This is actually a paper we published last year in Nature Digital Medicine, which highlights how we can use artificial intelligence approaches to automatically identify these biomarkers. But the next step is really not only using artificial intelligence to find these biomarkers, but it can actually identify new risk factors, maybe even allowing these AI-based tools to predict which patients are going to convert to advanced macular degeneration, how fast someone's atrophy is going to enlarge. You might imagine that being able to do these automatic predictions could be quite useful for identifying patients who may most benefit from treatments, this so-called era of personalized medicine. So to conclude, macular degeneration is certainly an important cause of blindness in the US. It can result in vision loss from new blood vessels or atrophy. Atrophy is really the big unmet need because we have tools to control wet AMD. And Dr. Ladd will talk about some advances in our treatment potentially for atrophy. But ultimately, I think in the future, we'll want to treat as early as possible. And these biomarkers will be very important for identifying patients who are at risk. And artificial intelligence, I think, will really help us in doing this. So with that, I thank you for your attention. I'll stop and turn things back over to Ramiro.
Thanks so much, Buzz. So our next speaker is Dr. Ladd. She will give a presentation on dry AMD from biomarkers to therapies in the pipeline. Dr. Ladd is a clinical scientist and retinal ophthalmologist with the primary goal of developing novel strategies for early diagnosis and treatment of AMD. Dr. Ladd's academic career goal is to translate her doctoral training in neurodegenerative disease into developing innovative diagnostics and therapeutic approaches for AMD. Her research interests are centered on biomarkers for dry AMD diagnosis and progression, and includes visual functional testing in dry AMD, investigating the role of new inflammatory AMD pathogenesis, and elucidating the use of novel retina imaging biomarkers for diagnosis and progression of AMD. Dr. Ladd has served as Director of Clinical Research at Duke Eye Center, primary investigator for investigator-initiated and phase 1 through phase 3 clinical trials at Duke, and as Director of Grading for Retina Clinical Trials at Duke Reading Center. She has fulfilled the role of International Lead PI for the Apellis Phase 3 OAK study for geographic atrophy secondary to AMD. Dr. Ladd collaborates with industry sponsors on development of novel, approvable visual function assessments in the context of clinical trials for retinal disease, with particular expertise in dry AMD, both intermediate and GA. Nora?
Hello, thank you so much, Ramiro, for the introduction and all the great work that you and Apellis team have done to develop therapies for our dry AMD patients. So I'm very honored to be here today as part of this wonderful event, perfectly timed during the AMD Awareness Month. It's a pleasure to join you and to speak to you today about the path from various disease biomarkers to current therapies in the pipeline. The work that I'll talk about today, importantly, has been generously funded by Research to Prevent Blindness, as you heard, National Institute for Health, VA, and through research collaborations with industry sponsors, including Apellis, all of who are very invested to improve the standard of care of our AMD patients. So the dry form of AMD, as you have heard from Dr. Sada, is a disease that ultimately robs the central vision. It's been very difficult to tackle. Therefore, the quest for markers of progression and future treatment response is very, very important. The biomarkers of dry AMD come in 3 flavors, can be divided into these 3 important categories: functional, structural, and blood-based. Dr. Sada has beautifully detailed the spectrum of AMD, the need for early intervention, and structural biomarkers, including AI strategies. So instead, I'll mainly speak to you about the first and the latter category. Functional biomarkers are interesting because the regulatory agencies of the Federal Drug Administration and European medicine agencies favor them. There is a strong reason why we would work on investigating functional biomarkers for dry AMD. Patients in the early intermediate stages, although they might have unaffected visual acuity on our standard visual acuity charts, experience certain visual function deficits. In the case that you see on the right of an 81-year-old patient, she has 20/20 vision on our charts. However, she has a lot of trouble transitioning from light to dim light and has severe loss of contrast, which is very bothersome to her because she's an artist. Future research is indeed needed to evaluate these functional impairments, which can then serve as future endpoints for clinical trials in dry AMD. There are a variety of visual function tests that have been investigated for risk of progression of the earlier stages of AMD. From left to right, you can see best corrected visual acuity, then low luminance visual acuity, which is best corrected visual acuity while darkening the vision of the patient with neutral density filter that decreases luminance. Then we have color vision that can be measured through various methods, contrast sensitivity, and we can, macular sensitivity using a test called microperimetry. Dark adaptation is also a test, the latest diagram, that tests rod function, and the rods are cells known to degenerate first in AMD. So at Duke, we started a natural history study named the Duke Feature Study, which followed the pilot study initially funded by the National Eye Institute. And then we did a longer longitudinal study performed in collaboration with Roche Genentech which had the goal to identify reliable functional endpoints and evaluate predictors of AMD progression and visual acuity loss in patients with early and intermediate AMD over 24 months. Low luminance acuity vision, cone contrast test, which is the color vision test, dark adaptation, and microperimetry were identified as markers of intermediate AMD and differentiated these patients from normal controls. At 24 months, we saw that cone contrast testing, dark adaptation, and microperimetry showed a functional decline in patients with intermediate AMD. Similar results were obtained recently by another international investigator, Dr. Robin Geimer in Australia. In a recent systematic review that we also did, we compiled a summary of all categories of dry AMD biomarkers. What we did was we screened over 1,000 studies, found 94 that qualified and had all our search criteria. And we found robust data that suggested the functional biomarker and testing protocols need to be tailored for any AMD disease stage. Specifically for early AMD, dark adaptation works best. For intermediate AMD, macular sensitivity using microperimetry and low luminance visual acuity. Lastly, AI or deep learning and combinational algorithms analyzing multiple types of biomarkers for dry AMD showed the greatest promise in assessing risk factors, detecting disease progression, and potentially predicting patient response to dry AMD therapies. So in addition to the visual function endpoints, which have been, again, favored by the regulators traditionally, there's some promising retinal images biomarkers that predict progression to geographic atrophy and wet AMD, and they have been recently described. Dr. Sada beautifully summarized them in his presentation, so I'll not go over them in detail. Suffices to say that with the advent of high-resolution retinal imaging of various types, especially optical coherence tomography, we're learning more and more about potential predictors, risk factors for disease progression in AMD. So you may ask, okay, but what about the relationship between structure and function? And indeed, the correlation between the two is very meaningful, especially in the earlier stages of AMD. So at our institution, Dr. Sina Farsi and Cindy Toth overlaid drusen maps and retinal pigment epithelial maps with the location of decreased retinal function in patients. These were patients enrolled in a natural history study done at University of Alabama, co-led by Dr. Christine Curcio, a prior speaker at this Lunch and Learn event, and Dr. Cynthia Ousley. The green disc and the white dots represent the spots in the retina where function was tested with dark adaptation and perimetry. Our collaborative group showed that in patients with dry AMD, worse cone sensitivity and slower dark adaptations to slow down rod function were related to structural markers on OCT. There was another study, a pilot study done by the National Eye Institute called the AMD Genotype and Phenotype Study. And we've shown the retinal sensitivities at individual microperimetry loci depicted on the left correlated inversely with the underlying drusen volume. So basically, thick drusen in AMD diminished the function over overlaying retina in this specific area. In addition to the work on structure function correlations in natural history studies of AMD, I'd like to briefly mention another disease which informed what we might want to pursue in the future for AMD. This is macular telangiectasia type 2 or MacTel. So what we did here, and first I'd like to introduce briefly the disease. So top left, the darkened area represents photoreceptor loss in this disease that affects the area temporal to the fovea. And Dr. Farsu developed the semi-automatic software at our institution to register the ENFOS OCT, which you see ENFOS images here in yellow and red with the green microperimetry maps. And he was successful in doing so despite the great differences between the two modalities. On the overlay of the two types of images, you can notice that the vessels are completely overlaid. And the images were beautiful. You'd argue they reminded me of Rothko paintings. However, what we found most beautiful was the fact that even when you went to the 3D, three-dimensional OCT, the overlay was nearly perfect. So what we did next, we analyzed individual overlays from patients enrolled in the phase 2 trial of MacTel of NTMT02. It's a CNTF implant. And we found that there was a nearly perfect correlation between structure and function. And these results were the basis of the FDA accepting photoreceptor loss on OCT for the first time as a primary endpoint for the current phase 3 study for MacTel patients. So this type of approach might be helpful to AMD because we're able to learn here that structural biomarkers can serve as surrogates for function. And we all know that retinal images or structure is faster. It's also more objective and convenient for patients than functional testing that can take a while. So because of the distinct advantage of the structural biomarkers, we wanted to learn even more by using retinal images directly via deep learning. I will not go into AI in detail because this topic has been covered by Dr. Sada. But what I like about this method is that it's not based and biased by our prior medical knowledge as much. So it might have the potential to uncover valuable new information about the disease. Importantly, it does not require the very expensive and labor-intensive human grading. So the algorithms created here can be used in large data sets. So at our institution, Dr. Larry Karen and Ricardo Henao developed a deep learning algorithm with excellent performance that predicted with high accuracy what patients will develop geographic atrophy in 1 year from intermediate AMD. And what was really neat to us is that the algorithm was able to generate attention maps for OCT volumes for eyes with intermediate AMD that will develop GA in 1 year. The images here are paired. These are pairs of patients. So on top, you have the attention maps of the patients with intermediate AMD. The red dots highlight the areas that the model identifies to be most predictive of future geographic atrophy. So this was exciting to us because the AI model no longer was a black box, as most of them are, and now we understood what biomarkers the algorithm was paying attention to on its way to GA prediction. Now, some of the highlighted features were consistent with prior literature and have been described prior work by Dr. Sada, and some were a total surprise and were unexpected and have not been previously identified by human experts. But maybe this shouldn't surprise us. Computers are known to solve problems in unexpected and seemingly counterintuitive ways. When the best-known world chess champion, Garry Kasparov, lost for the first time to Deep Blue 97, the computer's AI program finally was able to surpass the human expert's computational abilities. It finally mastered what's called the type A and B skills, high computational power to examine every single possibility to find the best move, and through programming of prior successful games. As Kasparov described in his book in 2017, Deep Blue performed very non-human, counterintuitive, high-risk moves to problem solve and win the game. So perhaps it's not a surprise to us that our algorithm identified novel anatomical findings not previously described. The biologic basis for these findings remains to be elucidated by future research by human experts. The third category of biomarkers is also very interesting. They're molecular markers. Our group has demonstrated that specific monocyte markers and subsets from patients' blood samples have been associated with intermediate AMD. You can see these data on the left. This work was generously funded by the Research to Prevent Blindness Special Scholars Award and led to a follow-on of the Merit Award. On the right, you can see another study done by Dr. Robin Geimer's team that showed that phagocytosis, or engulfment by macrophages, was reduced in all subsets in patients with intermediate or late AMD. This might lead to accumulation of debris and signs characteristic of dry AMD. So we wanted to learn more and look closely at these monocytes and macrophages. So we did some histopathology work in postmortem eyes, and we observed high numbers of macrophages stained here in red on the right in the outer retina subretinal space along the edges of GA mid-drusen. These data suggested to us that these cells are critically involved in disease process and progression. So this work was also funded by Research to Prevent Blindness and by Career Development Award through the NEI. Going back to the Duke feature study, we also looked at some genetic variants that were genotype 9 in numbers, and we found that the genetic burden score correlated with the disease stage and moderately with dark adaptation. There were 4 suggestive associations between vision function variables and specific genes. These classes of biomarkers paved the way for development of future therapies for our patients. Unfortunately, so far to date, the only FDA-approved therapeutic option for dry AMD are the REDS2 vitamins seen here. They only slow progression to the wet form by 20%. So we need we need to develop new therapies for this huge area of unmet need. So fortunately, in the past couple of years, we experienced a genuine explosion of therapies in the pipeline. On the left, you can see a list of strategies in development. The first half or so in blue are drugs, also visually illustrated on the right in terms of pathways approach. The bottom half lists devices or drug-device combinations. The complement cascade has been shown to be critically involved in AMD. And on the lower right, you can see that the drusen contain these spherules, which are composed of amyloid, the protein that deposits in the brain of Alzheimer's patients, and the anti-inflammatory proteins complement. Although the large anti-amyloid trials have failed, there are earlier approaches that target the more toxic oligomeric species. However, the most advanced clinical trial program in dry AMD are targeting the complement cascade depicted here in the main diagram. There are currently 9 large phase 2 or 3 trials of anti-complement therapies currently ongoing. They're performed either via intravitreal, subcutaneous, oral, or gene therapy approaches. Of these, the most advanced complement program is the Apellis phase 3 trials, and also the Iveric Bio's phase 2-3. Both are using the intravitreal anti-complement molecules. The OAKS trial by Apellis is the first large phase 3 trial that recently demonstrated success in RGA patients. Environmental exposures to Western lifestyle also predisposes individuals to AMD and perhaps to inflammation. So these factors are cigarette smoking, Western diet, pollution, all of which decrease function of the mitochondria, which are the energy-producing organelle in the cells. Antioxidant drugs are addressing the environmental problem, and they're administered via subcutaneous or intravitreal approaches, and they're developed by Stealth and Allegro. Other drugs in development are high temperature requirement A1 enzyme implicated in the progression of AMD. It has a compelling genetic association. It's being studied currently in a phase 2 trial by Roche. There's a synthetic oral DHA and also an intravitreal small interfering RNA, both in early stages of drug development. Next, I'd like to speak to you briefly about devices. We use a lot of lasers in retinal work, and they work well in a variety of retinal conditions, but have not been successful in geographic atrophy trials. However, in contrast, on the left, photobiomodulation is being used. It uses low-level light therapy with visible or near infrared light to stimulate cellular, likely also mitochondrial functions. This technology is being investigated in phase 2 studies. On the right, optogenetics, a very promising strategy to stimulate retinal neurons with light, is being employed by 4 companies in the GA space. Next is cell therapy, also very exciting. Can use cells from a variety of sources implanted through a number of modalities. And then on the right, retinal prosthesis, Argus 2 and the Prima system have completed safety studies in GA patients and showed evidence of efficacy following successful placement under the retina. So with these therapies, I would like to end on an optimistic note. Despite some hurdles with research in dry AMD, it is moving forward and making a lot of progress. Science, animal models, which I haven't had a chance to talk to you about today, biomarkers of all these categories have improved, leading to a genuine explosion of new clinical trials, as you've seen here. Some have failed in the past, but a myriad others are underway. And the new imaging modalities, visual function, and blood biomarkers will be very helpful to us to target enrollment of the right patients and showing success. So the work I showed you here from our group was done with the help of multiple collaborators that contributed and funding agencies. Special thanks to Research to Prevent Blindness for funding the early work on biomarkers and for the invitation to speak today, and to all of you for being here today in support of AMD research.
Thanks, Nora. That was an excellent talk. So maybe I have a first question to both Vas and Nora. So today, when you see a patient in your clinic with GA, how is your consultation with them in terms of trying to assess what's going to happen in the future? But then also when we think about things that are coming both from imaging technology of disease progression, but also new therapy, how do you see that changing over time? So maybe Vas, can you start with that?
Yeah, thanks, Ramiro. I think, as Nora beautifully highlighted there, I think there's real hope now for patients who have atrophy. That's really been kind of the biggest challenge for us since we've had treatments for wet AMD. And so one, I think, is that it's important. An important part of our discussion for patients is the fact that there may be opportunities to have the treatments to try to at least slow down disease progression going forward in the future. But for a particular patient, as we tried to highlight in these presentations, we really are trying to find patients who may be able to benefit the most from treatments and who may be in most need of them. And in terms of the important considerations, one is you kind of do want to know how fast things are progressing in a particular patient situation. One thing that we do know is what's happened in the past is a great predictor of what's going to happen in the future. So understand, being able to see patients over many years, in some cases, I have a pretty good idea of what's happening in a particular one of my patients' eyes. And so I think that's going to be helpful. Nora highlighted some of the functional issues, because ultimately it really is about patients' vision and their function and how they're doing in daily life. And how are both eyes doing? So we take all those things into consideration. So I'm trying to get to better assess what's happening in my particular patient, and then the other big aspect in counseling patients, I think, is talking about the future and what to expect.
Thanks, Faz. Anything to add, Nora, on advice for a patient that comes to your clinic today with GA?
No, I couldn't agree more. I have a very similar approach. I evaluate where they are today, whether the center of the vision is involved or not. And I tell them there's a huge pipeline currently in development and that there is hope. First of all, that's an important message. And the second is there'll be clinical trials that would fit certain patients best, as Vas mentioned. So, and we try to enroll them in the studies that we have ongoing at our institution. Sometimes we refer them around the country or even the world if we don't have specific studies. So we try to help in every way we can. We also refer them to vision rehabilitation or low vision. We have really talented occupational therapists that work with them on improving their vision through visual aids.
That's great. So we have a few questions from the chat. And the first one is about information on things that can prevent the occurrence of AMD, especially in terms of lifestyle and nutrition. I think, Nora, you talk about vitamins and minerals. So if you could talk a little bit about things that we can change or at least decrease the chance of developing AMD and GA.
So I speak to my patients about healthy lifestyle, healthy diet, green leafy vegetables and fish. And I tell you, if you don't like those, or you cannot use green vegetables for a variety of health conditions, then I tell them take your vitamins, the AREDS2 vitamins, if clinically indicated, if they have the intermediate high-risk drusen stage. For the early stage, we prescribe a general multivitamin, sunglasses and a hat, because the UV rays are detrimental to the back of the eye. Avoidance of smoking. I record whether they smoked or not, for how long, etc. I talk to them about family history and genetic risk for it. And what else? I'm sure I'm forgetting some things here. And there are a lot of resources online that we can provide to our patients. We have brochures, pamphlets. There's an AMD cookbook as well that's very helpful for patients. AMSA grid versus 4C home is also very important to prevent, to detect the earliest converters to the wet form of macular degeneration.
And for in your clinic, for patients that come that are concerned that either their parents or somebody from their family developed dry AMD, today are you using one of those genetic tests to investigate if the patients are at high risk of developing AMD?
I do have some patients that have had genetic testing performed that they come in concerned about the alleles that were discovered to be associated with AMD. So I do a full exam and reassure them best I can. I tell them that we recommend the full dilated exam once a year, even if there's no signs of AMD at that time. And even if they're younger, I tell them the definition of AMD, which includes age. So if they're younger than 50, it'd be very low likelihood. So that further reassures them, but I still recommend the full dilated exam and all the precautions, healthy diet, exercise. Your primary care doctor is also, they have to control blood pressure, cholesterol. Sunglasses and a hat.
So Vas, a question to you from the audience. When we think about clinical endpoints in clinical trials for geographic atrophy, I think that we know that those are major hurdles. So as a research community, how do you see that overcoming these hurdles of clinical endpoints for geographic atrophy?
So yeah, thanks, Ramiro. I think it's an excellent question from the audience, and that's where I think this biomarker research is very important, because as I tried to highlight in my presentation, we're really trying to understand the progression of the disease, what are factors associated with more rapid progression, and what are the stages of progression, because ultimately we would like to take these promising therapeutics and go earlier in the disease process. And when you have a disease that sometimes can take many years to progress, you really need to have this greater granularity in terms of understanding the stages of disease. In terms of how to get there, I think that's one of the things that we've been doing, obviously, is in trying to aggregate all of the knowledge, establish consensus groups to talk about how we can parse through these different stages in disease progression. But the biggest thing, of course, is really data. And how do you collect this vital data using all of the great tools that we have? We talked about a lot of these different imaging techniques, but you really need multimodal imaging and collected over periods of time at frequent intervals on patients. There are efforts. There's the Macular program, for example, in the EU that's trying to collect such data, but there's also a lot of data that exists in all of our clinics on patients that we followed over many years. And one of the efforts that we're looking at right now to try to do is to aggregate these large data sets, take advantage of artificial intelligence tools to be able to probe them. And that will help us, I think, refine the staging of macular degeneration into a very fine and granular fashion, such that we would be able to have new endpoints that are at earlier stages, as well as have multiple endpoints that we can track over time that could be used potentially in the context of new therapeutics. So I think that work is in progress, but it really is, I think, these types of analyses and development of more granular staging approaches that are going to be key.
Thanks, Vas. And then, Nora, when we look at more on the early stage of drug development and the use of animal models, anything that you see that has been useful for dry AMD in the development of new hypotheses or new drugs?
Animal models have been difficult. They are useful in replicating specific pathways of the disease. A good animal model of AMD, similar to a good animal model of a neurodegenerative disease, is very difficult to create. And in AMD, it requires all the components that lead to AMD in humans: aging, high fat, high cholesterol diet, and genetic manipulation. These animal models are very expensive to create. For example, at our institution, Dr. Bose Rickman has created a good animal model of AMD, but it requires years to make. It's very expensive to maintain those colonies. So I think animal models are useful in asking specific questions related to specific pathways of pathogenesis to AMD. So definitely valuable. In some studies, because of a holistic and comprehensive nature of the disease, pathology data has been very important too. So looking at human samples, that has its own limitations because of how you fix human postmortem eyes. But I think it obviously recapitulates the human life model closest. And then there's cell cultures that also, in which you can also ask questions related to specific pathways and get the answer much quicker.
Wonderful. First, we have one last question here in terms of AMD affecting certain photoreceptors, right? So why the macula, and then in GA, we know that the majority of the patients, they start with the atrophy on the extrafoveal lesion. So is there a reason why AMD, the macula, and then why GA is extrafoveal?
Yeah, that's such a great question. It's kind of at the tip of the spear in terms of research and understanding why that should be. I think there's some ideas, but certainly no definite answers. The macula, as I highlighted and sort of in the very basic introduction I gave, is a very specialized area in the sense that it is the highest resolution area of the retina. And and I use that analogy of sort of a multi-megapixel camera in the center, it really is very apropos because there is a tighter packing density of those light-sensitive cells. And so you can imagine that the metabolic needs of the macula are going to be substantially greater than other parts of the retina where there may be less density of cells. It's kind of like the highest population center, if you will. And when you're in high population centers, I guess maybe that the analogy applies in various parts of the world as well, you have other problems. And so some people believe that these drusen deposits and other materials is almost like trash building up in the eye. And so you can imagine if you're in a particularly metabolically active area, which has this high packing density, and maybe with aging, you might have impacts on your vasculature and other deficiencies that might develop in our cells to be able to handle this high metabolic load and processing over time, whether it's impairment of the mitochondria, the energy factories in our cells. All of those things can manifest in areas that may be most susceptible, and that might be in that most metabolically active area. But again, the topic of great interest and study, and as Nora highlighted, there's a challenge in animal models because aside from primates, animals don't have maculas. This kind of specialized area. And that's one of the reasons why we don't even know more at this current stage.
Yeah, wonderful. Vas, Nora, I think those were, we have maybe one last question here, Brian, if we have time. So a question to Vas, can you comment on the importance of stabilizing mitochondria in RPE in treating dry AMD or GA?
Yeah, so first, I should disclose a bias in that we have a lot of mitochondrial research happening at our institution. So I'm a big believer in the relevance of mitochondria. We certainly know there are age-associated alterations in mitochondrial performance and function. And so, yeah, I do think that the mitochondria are a huge part of why aging occurs in general. And so I do believe that there is merit in looking at that. And there obviously is, Nora also highlighted therapeutics that are even looking at targeting that. So I do think that that's important, but as I said, is an active area of exploration.
Wonderful.
So I was wondering if I could ask a question to each of our speakers. I'm wondering, in 30 seconds or less, could you tell us what you think is the single biggest limiting factor in terms of advances in this area of research?
I think that the biggest limiting factor, I think, right now is related to understanding all the disease mechanisms, right? Nora highlighted some of the therapeutics that are coming on board, which are based on rational information. We know, for example, when it comes to complement deposition and in those areas and genetics that would seem to suspect that. But the reality, the other thing is, Brian, we may be dealing with sort of not just necessarily a single disease, but maybe a constellation of different diseases. There may be many different mechanisms by which you ultimately can result in loss of these light-sensitive cells in the RPE. And so because of that, I think that's made it a little bit more difficult to unravel the mechanisms that may be in play at an individual patient. The other thing, Brian, that I often tell patients when I tell them, why don't we have a cure already? As I say, well, I tell them, when we cure aging, I'm pretty confident we'll cure AMD for sure then. So age-associated diseases are challenging to cure.
In my opinion, there are three major challenges with this disease. One is its complex nature, where 60% of it is a black box, despite all the outstanding research that's been done. And the second one is the difficulty coming up with the correct endpoints to treat the earlier stages. They're difficult. They should be functional, or you should have surrogate markers that are structured, that are really good. And the third is our lack of knowledge on what patients are best suited for enrollment in these studies. But the answer to all of those is biomarker and target development. So I think that's where we're all working hard, and we'll keep chipping away at this very complex multifactorial disease. That's a challenge.
Well, thank you. And we're out of time. I want to thank Dr. Ribeiro for moderating very ably and for great presentations from Dr. Basada and Dr. Noralad. Thank you all. It was really a very, very great session and really gives a lot of hope for the future in terms of some of the advances that are being made. Thank you.